This is the second video about a paper I helped to write. In the last video, I introduced the authors. I pointed out that ionizing radiation causes cytokine storm, just the same as the path to acute respiratory distress syndrome in COVID-19 involves a cytokine storm. Ketone ester was able to save more than 95% of mice given an LD70 dose of radiation. This led us to take on the task of looking at the potential to treat COVID-19 with metabolic therapy. I discussed the best time to treat COVID-19 with ketone ester. As in radiation, it would be important to start the intervention as soon as possible. I suggested indications for taking ketone ester include a positive COVID-19 test and elevated C-reactive protein. In this video, I want to talk about 4 roles of type 2 alveolar epithelial cells. Figure 1 shows a SARS-CoV-2 virus. The virus has a spike protein that attaches to the angiotensin-converting enzyme type 2. This cell membrane protein appears in many organs. The virus could infect cells in the gastrointestinal tract, the kidney, or lung. They all have the angiotensin-converting enzyme. Cells in our body that have a nucleus have mechanisms built in to detect a viral infection. The response of the infected cells to detecting a virus begins with the cell releasing interferon. This interferon will start a cascade of processes that eventually leads to an acquired immune response that will clear the virus. Inside the lung, there are finer structures called alveoli that are lined by alveolar epithelial cells type 1 and 2. Most of the inner surface is lined with the squamous type 1 cells. This is where the gas exchange happens. One also finds some native macrophages in place before the infection. The cuboidal type 2 alveolar epithelial cells are targeted by the SARS-CoV-2 spike protein because the type 2 alveolar epithelial cells have the angiotensin-converting enzyme 2 protein that provides a docking motif for the virus to enter these cells. There are 4 roles that could get wiped out by the death or impairment of these type 2 pulmonary epithelial cells due to infection with the virus. The roles include making surfactant, differentiating into AEC type 1 cells in healing, releasing superoxide dismutase 3, and releasing catalase. Let's take a look at each of these. What does surfactant do? We know that infants develop the ability to produce surfactants late in gestation. Babies born prematurely did not survive. Today, these children are treated with exogenous surfactants and their survival rates have greatly increased. Surfactant lowers the surface tension in water. Surface tension is used to describe many phenomena. How does capillary action work? Why are raindrops round? How can insects walk on water? What keeps the alveoli from filling up with water? Surfactant produced by the alveolar epithelial type 2 cells is a mixture of lipids and protein. The main lipid is dipalmitoyl phosphatidylcholine. There are other fatty acids than palmitic acid in phosphatidylcholines. There are also phosphatidylglycerols and phosphatidylinositols and cholesterol present. These form a monolayer at the water-air interface, with the hydrophobic portion pointing towards the air and the hydrophilic portion facing the water. The effect of the surfactant is to lower the surface tension. Surfactant is roughly 90% lipids and 10% protein. The 4 proteins, surfactant protein type A, B, C, and D, are also produced by the type 2 alveolar epithelial cells. Surfactant proteins A and D interact with macrophages, enabling phagocytosis of viruses and bacteria. Surfactant proteins B and C are necessary for positioning and maintaining the lipid components of surfactant in the air-water interface. As part of the healing process, the AEC I cells will need to be replaced. This requires AEC II cells to divide and differentiate into AEC I cells. A lack of AEC II cells in the alveoli or inability to undergo mitosis and differentiate into AEC I cells would doom the alveoli to being filled with scar tissue. These AEC II cells also produce superoxide dismutase 3 and catalase that are released from the cell. It is worth spending some time on understanding SOD3 and catalase. That will be the topic of the next video.